们现在要去 Rpi 的实验室去参观。它这些楼都是后来建的，还挺现代化的。
Influence from where it comes from. And so if there's any questions on this topic, I'd be happy to answer. Cool. Not cool. Not cool that there's no questions. But if there's no questions, all right. Give up my sci-fi roots. Why I'm here, happens in Star Trek. And so, I think one of the very interesting things that we're seeing in the research funding space that isn't getting well publicized, isn't politicized, is how the lunar economy is shaping scientific research right now, as funded by NASA. You might ask yourself, why? And why would someone look at, this, you don't have to really read all of this, I mean, if you want to, go for it. But, it's all about extremophile research, understanding how biological processes occur in these really extreme environments. And it's all for producing food, producing medicine, producing whatever human beings need to explore space, to be on the surface of the moon. And so, Dr. Blaber, this version is left, we have another version of it, but merging, the ability to look at like the structural genomics of molecules using genetics and advanced imaging, advanced data collection.
And they said, don't stay.
We are connected we can be connected to a quantum. And flies and fish. No canines. So these are incubators. A lot of times you might use those centrifuges like I was just talking about, and then you take your samples and you might put them into a temperature-controlled space. These things shake. A lot of times what you need to do with biological elements is agitate them, move them around. That in and of itself, we talked a little bit about thermodynamics a second ago. That creates a thermodynamic event, the shaking, in fact. And so it's a very mechanical, but, and I think that's kind of exciting in and of itself. We have far more Amos, our supercomputer. I would say half of our labs are literally connected to the Amos system here. And so what they do is they use things like our NMR, this big device over here, nuclear magnetic resonance machine, to collect information and use computers to solve computational problems with it. In fact, we have one of the leading reasons, probably one of the top, protein thermodynamics. That's a really esoteric field. We have the leading researcher on the planet working here. Listen, granted, it's a small space, but he knows more about the thermodynamics of proteins than anybody else in the world. And so, what do you do with that? Well, you build other projects around it. You use it as you're developing new molecules, as you're developing an understanding of enzymatic reactions inside of a bioreactor that would otherwise be taking like sequenced inline steps through a chemical plant. The hardest thing about making bioplastics right now is the fact that you have to make them in a vessel that's about 1 million liters if you want to make them at scale. And all bets are off after that. You don't know, you can't predict what you did in a 10,000 liter reactor will happen in a 1 million liter reactor. And the cost of failure is so incredibly high. So we need a brand new way to look at things, meaning brand new biophotonic sensors, brand new systems, brand new algorithms for just how to express whatever it is you're doing in that bioreactor into a more traditional system. So we're continuing to get people, that's awesome. Any questions before, because what we're about to, high heat dishwashers, autoclaves, if anyone is familiar with autoclaves. But they, you know, the water temperature is hit, I think ours are capped at about 260, but they can go much hotter. Obviously, you know, you're processing elements that need higher heat to destroy, that would be used. But in this case, we have nothing that you can't kill with 260. So you can walk on through. And so through this bioart project, we're demonstrating that there are certain microbes in the runoff of this mud that produce, biochemically produce pigments that are literally all toxic elements are stripped out. And you're just left with maybe not as blue of a blue. Really blue is hard to make, it's expensive, and it's also gross from a production standpoint. And it's but getting a royal blue is a tough target to hit. But through this very sort of, you know, bioart oriented experiment, you're starting to see where the confluence of, you know, biomaterials for textiles, material science for colors, and art come in a confluence demonstration. And so what am I approving for the next tour? Is a mud sculpture that dynamically changes its color over 2-3 weeks of time. And we'll have a time-lapse camera taking its, showing this. And